Welcome to Let's Talk Micro. Hello everyone, welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Google Podcasts, TuneIn Radio, Good Pods. Wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on LinkedIn as Luis Plaza, on X as Let's Talk Micro 1, and I have an email address which is letstalkmicro@outlook.com. So please subscribe to the podcast, download episodes, leave a review if the app allows you to do so. And if you have any feedback, any topic suggestions, please go ahead and send those via social media or via email. Any feedback, any suggestions, they are always welcome and appreciated. And if you haven't listened to the previous episode of Let's Talk Micro, please go ahead and do so. It was a great episode about Marburg virus with Dr. Caterina Miraglia. And Dr. Miraglia, she's a professor. She teaches microbiology at the University of Massachusetts, Dartmouth. And her background is she is a medical laboratory scientist. And she published a review article in 2019 about Marburg viruses. And she did that because, she saw what was going on with Ebola, and she wanted to make sure that her fellow medical laboratory scientists, they had an understanding about these type of viruses. So I really applaud the effort about making sure that we're all on the same page, providing information. It is always great. As I always said in this podcast, sometimes, we get overwhelmed by information and we're so busy with our work and, trying to find articles here and there or listening to the news or reading articles. So it's always nice with someone in the profession. They take the initiative and they do something like this. So we talked about the virus, as always, breaking it down. With viruses, you talk about things like DNA or RNA, mode of entry, transmission, any prevention, treatment. So it was a great episode. So definitely, if you haven't checked it out, please go ahead and do so. So in today's episode, I am going over a topic that I covered about 2 years ago, which is MALDI-TOF MS. And I am covering it again for some reasons, If you listen to this podcast, you know that recently I did a webinar. Maybe you saw me promoting it through social media. So I did a webinar for the American Society for Clinical Laboratory Science, which is ASCLS, about MALDI-TOF. So it was good to talk, about it from the point of view of a medical laboratory scientist, to go over the components. That way everyone in the community is familiar with it. Maybe you might not be too familiar outside of microbiology. You might not know what it is. So I did that webinar to talk to the medical laboratory scientists and anyone that wanted to sign up for it, so they could get a better understanding about what is MALDI-TOF MS and, how is it used in the lab, how it has helped us, and at the same time, with advantages, what type of advantages it has brought, and also what type of disadvantages it has brought. So I think it had a great reception, it had great feedback. And since by now this podcast has more followers than it used to, I thought it will be a good time to kind of just talk about it again. That way new listeners, can benefit from this. Maybe you're a new medical laboratory scientist, a recent graduate, you're a student, or you might not be familiar with MALDI-TOF MS at all. So I think now is a good time to talk about it again. Like I said, now Let's Talk Micro has new followers. Maybe you're just listening to the recent episodes. You haven't gone back to the old content. So, and you're upcoming in the community, you're recently starting to study medical laboratory sciences or microbiology. I think this can benefit everyone, not only the medical laboratory scientists. So let's go ahead and talk about MALDI-TOF MS. So what I'm going to be doing is, in this episode, I'm going to be going over the components, how it works, how it helps the lab, talk a little bit about how things are done in clinical micro, how the workflow is and how cultures are read, and how MALDI-TOF applies to the to the work that we do in clinical microbiology. And then on a second episode, I will be talking about the advantages and disadvantages, so the pros and cons. So maybe you might have heard of MALDI-TOF MS, or you might not know about it if you're a medical laboratory scientist. And if you work in a large facility and you work in microbiology, you might work with it or might have worked with it. Or at the very least, maybe you heard about it. So definitely it's a trending topic. It has changed things, it has changed the way we do things in the laboratory. And this is a method that is used for the identification of microorganisms. It identifies bacteria, both aerobic and anaerobic, yeast, mycobacteria, and nocardia. And it reduces the time it takes to get an identification of an organism. So I thought it would be good to for people that work outside of clinical micro and you're listening to this, or if you're just getting started in a lab, I just wanted to go over a little bit on how the whole identification system works with traditional methods. So if you work in a clinical microbiology lab, you're familiar with this, and bear with me, so this is for the benefit of everyone. But as you know, in the lab we have systems and instruments, right? Like the Vitek or Microscan, and they have no relation to the podcast. And you will typically set up your sample in them, right? Some sort of suspension, and then you put like an identification card or some sort of test system, and then those will take hours, right? With Vitek, typically from anywhere from 4 to 8 hours, you can get an identification on an organism, and that is for bacteria. And then if we have yeast, it can take up to 24 hours. So you're basically setting those up, and then they will be read the next day, for the most part. And that is because the way that the microbiology lab is set up, it's a little bit different from other areas of the lab, like hematology or chemistry. Whereas, if you're a generalist, or if you have worked as a generalist, the person that's coming on the next shift, so if you're on first shift, the person coming on the evening shift, they come in at 2:30 p.m. or 3, they pretty much have the same training as you. So you're reading diffs, you're doing body fluid cell counts, you're doing a type and screen, you're doing a cross match, or you're doing some sort of chemistry test. The person that's coming to relieve you, they have the same training. So you can go ahead and, typically there's an overlap between shifts, like about 30 minutes. So at that time, when that person comes in, you can already have finished what you're working on. You start working on your pending log, and then that person takes over. They start reading slides, doing diffs, continuing blood bank testing, doing chemistry testing, etc. But that's not the way that it works in microbiology. The way that it is set up, it's a little bit different. So, for example, a lot of times, cultures, they're read primarily on first shift. Some hospital, they do have some rotation system in place where maybe like urine cultures are read on second and third shift. But other than that, sample cultures like wound cultures or respiratory cultures, they are read on first shift. So that means that any testing that you set up, the person coming in the next day will continue reading it. So that's something that's different. So going back to the systems, you have things like Vitek or Microscan, or you have some rapid testing where the test is, it takes about 4 hours. So people typically will set it up in the morning, and then maybe at lunchtime they will read it. And if you're setting it up later than that, you might have to maybe stay over, because once again, we go back to the person that's coming in on second shift. Maybe they're not trained in wound cultures, or they're not trained in respiratory culture, so they don't know how to read that test. So that's one area where micro, it's a little different from other areas of the lab. So you have these traditional methods that they already take hours. So basically, that ID is being released the next day. That is not the case with MALDI-TOF MS. The ID takes minutes, right? So that's something, if you're hearing it right, it's just great. You're getting an ID in minutes versus a day. So that's a big difference right there. And granted, I will talk more about the setup of MALDI-TOF MS. So when you say minutes, right, it's provided that it's depending on the amount of samples. Because when I go over the components, you'll see that if you set multiple samples and you have like a full target slide, and I will talk more about that, the whole thing, can take about an hour. But however, that is still faster than a day, right? So let's go ahead and talk about what is MALDI-TOF. So MALDI-TOF MS stands for Matrix Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. And it has several components. So it involves the use of mass spectrometry. And let's go back to our chemistry class, our chemistry courses. So mass spectrometry, it's a tool for measuring the charge to mass ratio of molecules. So in MALDI-TOF MS, you get a sample that is mixed with a matrix, and a matrix is a substance that ionizes the proteins. And then you use a laser to excite the chemical functional groups that are included in the proteins of an organism. And when the laser is applied, the matrix absorbs energy, transferring heat to the sample proteins and creating ions. So the matrix ionizes the molecules. The laser is providing the energy and desorption. And then those ions, they are separated on a tube that is referred to as the flight tube. The ions, they are measured using a detector. So the lighter the ions, the faster they will travel in the tube. And once they get to that detector, a mass spectrum is produced using a mass to charge ratio. And this spectrum is compared with known bacteria and fungal spectra that are stored in the database. And that's how identification is obtained. So let's go ahead and recap these components. So MALDI-TOF, Matrix Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. It uses mass spectrometry, and it has a time of flight tube. That's where your ions travel. You have a matrix that's added to a sample, and then that matrix is going to ionize that sample, which happens when the laser that's in the system, it points to that sample, and then you get energy, and then the matrix transfers energy and ionizes the molecules. And then those molecules travel through the tube, up the tube, to that ion detector, where a spectrum is created based on the mass to charge ratio. And you have a database of known organisms like like E. coli, Klebsiella, your gram-positive cocci, your yeast, and then that produces an identification. So now that you know what the components are and what MALDI-TOF MS stands for, let's go ahead and talk about how it works in more detail. And there are two main manufacturers that have no relation to this podcast for MALDI-TOF, which are the MALDI Biotyper from Bruker and the Bitec MS from bioMérieux. Once again, they have no relation to the podcast. So I'm more familiar with the Bitec MS, which is the one that I worked with. But both systems, they have the same principle, they have the same components, and that's what I'm going to be going over. So I mentioned that you have a sample that is mixed with a matrix. So let's start with basics. That sample it's placed on something that is called your target slide. And there are some variations with the manufacturer. And you hear some terms like acquisitions. For example, with the Bitec MS, your target slide has 3 acquisitions. And each acquisition you can put 16 samples in it. So that brings it to a total of 48. When you are testing the acquisitions, right? So the results are released after the testing is completed. So even if you get the spectra of one sample, if you have 5 samples, your samples, your identifications are going to be released when all 5 samples are done. And this type of systems, they communicate with the LIS, where you can get your IDs to be released to your LIS. So that works very well. And typically with the acquisitions, or that particular area, it's like a single time use. So you try not to be wasteful, and maybe if you have multiple samples, go ahead and set them up before you test them on the instrument. Because if you use it for one, then you cannot use that acquisition again. So you have a sample, you put it on something that is called your target slide. And then you go ahead and use the matrix, which, as I said, it ionizes the molecules, and we can get a little bit technical. But the matrix, it contains alpha cyano 4 hydroxycinnamic acid, or CHCA. So you might hear the word chica. And you apply this to your sample. And you also have a calibrator strain that you test along with your samples. And this is like an E. coli strain, and different procedures for the manufacturers. But the bottom line is that this calibrator strain has to pass. If not, the whole acquisition of tests fails. However, this can be an easy fix, where you can maybe reapply that calibrator and rerun your samples, because that happens for various reasons. Either maybe you didn't apply your calibrator, or you didn't properly put your sample on the target slide. And with the target slide, and you can definitely using a search engine, you can look it up. So it has some small circles, and that's where you actually gonna apply your sample. So, and this is done with a loop. You get a colony from your organism, and then you apply it to the sample, and then apply your matrix. And when you do that, you let it dry, and then you place it in the instrument. And with both manufacturers, you have some differences, which is that with Vitek MS, you use formic acid for yeast, and this is used for extraction. So what you would do is you get your sample, you apply it to the spot on the target slide, and then apply the formic acid, let it dry, and then apply your matrix. So with Bruker, you apply the formic acid on all the samples. So that's the difference. And I mentioned that you can, you apply your colony using a loop. There are some devices where you can pick the colony and then put it on the target slide. So there are some variations there. But you have to make sure that you have a good quality of sample, that it's a fresh sample, typically within 18 to 24 hours. Can be a little bit longer for yeast. Sometimes mucoid samples like Klebsiella, those can fail. They can fail to provide an ID. So you have to be careful how you apply those. And I found that with me, for me, it works that there's a recommendation that using a swab, you remove a little bit of the mucoidness of the colony, and then using a loop, spot your sample on the target slide. For me, it works that if I get a loop and I kind of wiggle it a little bit in the mucoid colony, and then I place my sample on the target slide, that works for me. You can also, and you hear the term spotting. That's when you're applying your sample to the target slide. So if I'm saying spotting, that's what I'm talking about. So you can also, spot it multiple times to make sure, that you get an ID. And that's when you have samples like that, like Klebsiella, they're mucoid. So depending on the system that you're using, once you have added your formic acid for all samples, if you're using Bruker, or you have added it for your yeast on the MS, on the Vitek MS, and then that has dried, and then you ultimately add your matrix, and that has dried, then you can go ahead and place your target slide in the instrument. So there now the sample has the matrix, so the laser is pointed at the sample, right? That energy, it's given by the laser. The matrix ionizes the molecules, and then the molecules travel up the time of flight tube, and then to the detector, and then a spectrum is produced, and that is compared to a database of known spectra. And like I said, while it is true that an ID is produced within minutes, if you're using a target slide, let's say that you fill all the acquisitions, ultimately, and maybe your sample is in the last acquisition, it can take like an hour for all those IDs to be completed, but it's still faster than other methods. Like any other system, right? Any test, every test, every system, they have some sort of limitations, and MALDI-TOF MS it's not the exception to this. And also with limitations, there are some factors that affect you getting a successful ID, and a very basic one is maybe failure to add the formic acid or failure to add the matrix. And those are easy fixes. You can just repeat it and then get the ID. Also, the quality of the sample, like I said, mucoid colonies, sometimes pinpoint colonies that you cannot get enough sample while you're spreading it. That can affect, that can prevent you from getting an ID. As far as but those are, quality of sample and user errors. But as far as the instrument, you cannot differentiate between organisms that are closely related, like E. coli and Shigella. So you will get it with a comment saying, you cannot differentiate between both of those, but you use your morphology and your biochemicals for that. And then those of you that are in microbiology or as MLS, MLS medical laboratory scientists, we went over this in school, but you have organisms that are closely related, and they are classified, they are put together in something called groups or complexes, like Enterobacter cloacae complex, that it has organisms like Enterobacter cloacae, Apsuri, Kobe, also Burkholderia cepacia complex. It cannot differentiate between Achromobacter psilocycens or the nitrificans, and also it cannot differentiate between subspecies of Streptococcus dysgalactiae. But those, like Achromobacter, you will have to do an ID by another method. With dysgalactiae, you can just release it as Streptococcus dysgalactiae. But when it comes to like the complex, which for the benefit of the audience, if you're just a student, or these are organisms that are closely related and they are put together in something called a complex or group. As I mentioned, Burkholderia, Enterobacter cloacae, so Burkholderia cepacia complex, Enterobacter cloacae complex. So in the cases that you get low discrimination between two organisms of that complex, you can just release the ID as complex. So if you get a low discrimination between Enterobacter cloacae and Apsuri, let's say, you can release that ID as Enterobacter cloacae complex. And the database of these instruments, the database of organisms, it's quite big. So more than likely you will get an identification, but doesn't mean that you will always get one. Even if you use a nice quality sample, you follow the procedure, you add your matrix, you might not get an identification, and you might have to either send your sample to another place if you don't have a way to identify that organism. So it happens. So even if you minimize user errors and you follow the procedure, sometimes some colonies they just won't identify, and that's common in the lab. So you will have to find another method. But overall, it's a great way to identify organisms. You get the identification faster, and definitely it has a lot of advantages and disadvantages. Unfortunately, not a lot, but there are some. Mostly in the way that it has changed the way that we work things and the thinking of technologies. And that is something that I will talk about in the next episode about MALDI-TOF MS. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about MALDI-TOF MS. As always, I enjoy sharing this information with you. If you listen to the series about MALDI-TOF 2 years ago. First, thank you for listening, and then thank you for listening again. I think this is a good time to talk about it again. We get, Let's Talk Micro gets new listeners. Maybe now you're going through school. Maybe now you're working in a lab. And sometimes, when we follow podcasts, maybe we don't check the older episodes. So this was 2 years ago. And this is a trending topic. So thank you for the support. Thank you for listening. And please continue bringing that passion to what you do. It's so important. You do such great work. So as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.